For cancer to be contagious, two hard conditions must be met. Have humans become so crazy now? I just made a few episodes about cancer, and friends messaged me asking if injecting cancer cells into a healthy person would cause cancer. What would happen if someone accidentally ingested cancer cells? Such questions are baffling, except perhaps for those with extraordinary imaginations. Who would dare think about injecting cancer cells into a healthy person, let alone actually doing it? Eating cancer cells, as if delicacies like dragon liver and phoenix marrow aren't enough to satisfy your taste buds. Now, people want to eat cancer cells? This matter can't be taken lightly. There's a serious side to it. Actually, scientists have conducted experiments on injecting cancer cells into healthy bodies and ingesting cancer cells. Those who were injected with cancer cells carcinogen is their ultimate fate. The tale should commence at the outset. Back in 1951, obstetrician Professor Richard Talinder took a tissue sample from a black woman named Henrietta Lacks, who had cervical cancer, and handed it to biologist Dr. George Gay for cultivation. Gay discovered that Lacks cancer cells could divide and proliferate endlessly outside the body, and within 20 to 24 hours, the number of cells in Lacks' tissue sample would double from 1 cell to 11 cells in just 30 days. This was a groundbreaking discovery, marking the first time humans had found cells that could divide limitlessly and are immortal. George Gay named them HeLa cells. On April 10, 1951, Gay announced this discovery to the world and generously shared HeLa cells with medical institutions globally. Thus, HeLa cells began proliferating in laboratories around the world, like bamboo shoots after a rain. Statistics show that over 70 years of their discovery, HeLa cells have replicated over 18,000 generations, weighing a total of 50,000 tons, equivalent to the combined body weight of 1 billion people. While HeLa cells were shared globally, some questioned their safety. In 1954, Edward State Cornell Medical College's virologist and oncology department head, Chester Southam, had a different view on HeLa cells. He raised the questions: many bacteria can spread among humans, so could these dreaded cancer cells be contagious too? What if scientists researching HeLa cells got infected? What would happen? Thus, a terrifying idea crossed his mind. Dr. Gay had previously injected HeLa cells into live rats, producing malignant tumors. So wouldn't humans develop tumors after being injected with HeLa cells? This thought wasn't unique to Southam. Many scientists believed cancer resulted from viral infections or immune system defects. What would happen if cancer cells were injected into humans? For scientists, exploring every unknown hypothesis holds scientific significance. Thus, Southam believed. Min injected HeLa cells into the patient's body, and he deliberately filled the needle with salt solution mixed with HeLa cells. A lady newly hospitalized with leukemia became Southam's first experimental target. He told female patients that they had developed a new drug which was used to test the immune system and would be of great help in treating leukemia. Female patients were thus involved in Southam's trial. Southam stuck a needle into the lady's arm. Then he slowly pushed the handle of the needle, and about 5 million HeLa cells entered the body of the woman. In the middle of the injection, a small bag bulged at the needle site, so Southam had to change the needle and continue to inject the lady. The rigorous Southam carefully pointed a small black dot on the side of the needle eye on the lady's arm so as to ignore it. After a few days, weeks, or even months, she can easily find the injection site and can better observe and check whether HeLa cells cause cancer, she thinks, an experimental subject. The available data are extremely limited, and their reliability is relatively low, so he used the same method to inject malignant tumor cells into the bodies of more than a dozen cancer patients. The reaction came quickly. Within hours of Southam injecting HeLa cells into the patient's body, the patient's arm began to show signs of mild redness and swelling. 5 to 10 days later, the patient developed a hard lump at the site where he was injected. That's what we often call cancerous nodules. Southam thought it might be a tumor, so he cut off a part of the patient's lump for testing. Surprisingly, these tissues are not part of the mass left by cancer cells, which are not removed for the patient, but to see if the patient's immune system can compete. If the cancer cells are not intercepted by the immune system, they will spread, and Southam estimates that within a few weeks the lump on the patient's arm will grow to a tumor 2 centimeters in diameter. almost the size of a coin, when Henrietta Lacks, the owner of Hella Hippo, underwent tumor resection at Johns Hopkins Hospital in 1951. The cancerous tissue on his uterus happened to be the same size. Fortunately, the heart lumps on most patients' arms disappear on their own within a few months after the injections. However, 4 vaccinators showed different symptoms. Instead of disappearing, their lumps showed signs of growth. Gertham could only surgically remove the mass for the vaccinator, but soon the mass recurred and eventually evolved into a tumor. For this reason, Southam kept removing them, but the tumor grew again and again, and one patient's cancer cells even spread to lymph nodes. Southam's operation was obviously the harm caused by these experimented subjects who knew the truth. But Southam's experimental results on his own patients were not satisfactory, because these patients inoculated with HeLa cells were cancer patients themselves. He also believes that a few patients are not enough to tell the problem. In order to better do the controlled injection tests, Southam targeted the vaccination on healthy people. In May 1956, when she participated in the experiment with Our Lady, Dr. Alice Moore took the prepared HeLa cells from New York to the prison in Ohio. The reason for Forehead Prison is that the warden here is a friend of Southam's. Besides, the prisoners here have been very cooperative with you in several scientific studies before. With the support of the warden, they started their work in this prison smoothly. First, he advertised in the Ohio State Prison newsletter, saying that 25 volunteers were needed to conduct cancer research with them, and the participants would be generously paid to reduce their sentences. The effectiveness of advertising is very great. In just a few days, 96 volunteers were recruited, with the highest number reaching 150. In the end, Southam selected only 65 volunteers who were guilty of murder, embezzlement of public funds, robbery, and forgery respectively. The selected people are arranged in a row, sitting on a wooden bench with their arms exposed, and then the doctor injects HeLa cells into their bodies. A few days later, the media also reported one after another. They called these prisoners who participated in the inoculation of HeLa cells heroes and praised them. It is the first healthy person in the world to volunteer to undergo such a rigorous cancer test. In the report, they quoted a prisoner, "If I said I was not afraid at all, it would be a lie." How do you feel when you lie in bed thinking about cancer cells on your arm? The reporter asked again, "Why did you sign up as a volunteer?" The prisoner replied that I have done something unforgivable to a girl, and I think I can always make up for it. Personally, I think these prisoners are involved in the driving force behind the inoculation of HeLa cells. It probably comes from the sentence in the advertisement that can reduce the sentence. After all, nothing in the world is more valuable than freedom. Soon after, the prisoners who were injected with HeLa cells showed the same symptoms on their arms as the previous hospital vaccinators, and tumors popped out of the arms of the prisoners who received HeLa cells injection. Obviously, these healthy vaccinators responded more slowly than those who were terminally ill in hospitals. Fortunately, however, these healthy vaccinators relied on their own immunity to defeat cancer cells. As a result, Southam injected cancer cells into their bodies many times, and the dose was increased every time. But strangely, these people injected larger and larger doses and produced smaller and smaller responses, as if they developed immunity. Moreover, the more injections their bodies engulf, and the faster the HeLa cells respond. Southam made his research public, and claims that this phenomenon provides great prospects for the development of anti-cancer vaccines. At the same time, it also seems to be a screening means. Some media in the United States began to hype that this research might bring a big breakthrough in anti-cancer vaccines. But this did not stop Southam's madness, but intensified it. He continued to use HeLa cells for the next few years, and other active cancer cells in human experiments. According to statistics, he has injected more than 600 people successively, half of whom are cancer patients, and the remaining 2/3 are other patients and gynecological patients. When patients ask Southam why, Southam always prevaricates with the reason of cancer testing. According to Southam's later account, patients who came to Sloan Kettering Memorial Hospital and James Ewing Hospital for gynecological surgery will be covered with cancer cells. Even when the vaccinator asked for the reason, he still simply said, "Conducting cancer tests." Because the bodies of cancer patients reject these cells more slowly than healthy people, Southam believes that by recording the rejection time, he could detect undiagnosed cancer. In addition to cancer cells, Southam injected West Nile virus into more than 100 patients in advanced stages of cancer. West Nile virus is a zoonotic infectious disease with severe consequences. It can cause fatal encephalitis in humans and horses and kill birds and chickens. Southam's work and subsequent research mainly focused on the role and pathogenesis of the actin West Nile virus, resulting in more than 10 published papers. Like the previous cancer cell injection trials, these studies were also published in the New York Times. causing a huge stir and reaction. However, honor was accompanied by those cancer patients also contracting West Nile fever. The year came to 1962 when Southam signed a secret agreement with a hospital treating chronic diseases. The agreement involved testing cancer cell injections on Alzheimer's patients in the hospital to determine whether these frail patients could reject cancer cells. But this deal eventually led him to the court and became a lifelong stain on his record. Some say that without this stain, he would have risen to the pinnacle of his career like a rocket. Who brought Southam to court? In 1963, the hospital's director summoned and ordered 3 physicians responsible for managing and treating 22 Alzheimer's patients to inject cancer cells. These 3 righteous doctors sternly refused and rebuked the director, but the 22 Alzheimer's patients did not escape the ordeal due to the attending doctor's justice. These 3 doctors were Avery Kagan, David Licht, and Perry Fusco. Academically, they were not well known, just ordinary doctors. One day, when the 3 doctors attended a hospital meeting, the 22 patients were injected with cancer cells and became experiment victims. Infuriated by the truth, the 3 doctors on August 27 submitted resignations accusing the hospital of unethical scientific research. They also reported Southam and the director to the federal court, citing violation of patients' informed consent and health rights, and conducting inhumane experiments on elderly patients who cannot express consent. The 3 doctors' report set off a media firestorm in the US, with criticisms against Southam for deceiving patients and violating medical ethics. Eventually, both Southam and the hospital director had their licenses revoked. Southam's experiments revealed that injecting cancer cells into a healthy person does not cause cancer. Why doesn't the injection of cancerous cells lead to cancer in different individuals? Wang Donglin, director of the oncology department at Chongqing University-affiliated Tuzhou Hospital, explains that for cancer to spread, 2 stringent conditions must be met. First, cancer cells must enter a healthy body. Second, they must survive and grow in the healthy body. Under normal circumstances, human tissues and cells are controlled by genes. The growth and proliferation of cells are orderly. Individual cells will break through the control of genes, become mutated cells, and eventually become cancerous. This is an internal reason and belongs to their own genetic mutations. The condition of cancer infection must be external cancer cells, and the external cancer cells want to enter the body of healthy people. In addition to the injection as we know above, the possibility of cancer cells entering the human body through daily diet, breathing, and contact is very small. I don't think anyone wants to eat cancer cells. Even if cancer cells successfully enter the body of healthy people, the possibility of survival is very slim, because the body's own immune system will detect the invader at the first time. The cancer cells entering the body of healthy people are then immediately slaughtered indiscriminately. It is unimaginably difficult to meet these 2 hard conditions, so cancer cells are not infectious. Even if you eat cancer cells by mistake, you will not be infected. But I can't describe the taste. After all, I haven't eaten it before. I think the taste of cancer cells should be similar to that of meat. What do you think? Okay, that's it for today's video. If you like my video too, please like me, favorite, comments, and forward. See you next time. Bye.